Chapter 1, Section 4, An Introduction to Microbial Life. Now we need to keep in mind, when you're thinking life here on planet Earth, pretty much all life falls into three categories, okay, three lineages. 2 prokaryote, 1 eukaryote. The 2 prokaryotes are bacteria and archaea. Eukaryotes, well, that's everything we normally see. Us, mammals, animals, insects, plants, fungi, protozoa, anything with a nucleus. Eukaryotes. Anything without a nucleus, without internal membranes forming organelles. Prokaryotes. The archaea. The bacteria. The bacteria, as we said in section 3 of this chapter, are relatively small, okay, compared to us, compared to our cells. I mean, you look at it, they're anywhere from 1/2 to, at best, 10 millionths of a meter long. There's about 80 plus phyla, okay? Why the plus? Why not an exact number? Well, because we're finding new ones all the time. Not only are we finding new species, and we're finding out new lineages of those species within the Linnaean, lineages. Because remember the Linnaean model of how to set stuff up, kingdom, phylum, kingdom, class, order, all of that stuff still applies to our friends, the prokaryotes. When we look at these guys, we have to keep in mind, there's those of us that are relatively visible, maybe with a magnifying glass or a very low-powered microscope. We can definitely see paramecium with a really good magnifying glass. You can see them really, decently with a dissecting scope that maybe does 40x magnification. We can see the yeast, we can see the diatoms. And we can see some of those giga bacteria we talked about the last section. But as we get smaller, we go from what we can see with light microscopes to things we now need to break out those lasers and use electron microscopes. Right in here, here's our friend E. coli. E. coli and many of the, what they call the bacteriophages, viruses that infect only bacteria. Some of the much larger viruses are going to be there straddling what we can make out with the light microscope and when we need to break out the electron microscope. We start getting down into subcellular organelles. Remember, organelles, that means us eukaryotes. Then we need to break out and really get to the transmitting electron microscope so we can see some of the much smaller viruses. The archaea, prokaryotes. There's 5 well-described phyla. Altogether 12-ish out there, but 5 we have really studied. I mean, these are the guys that are going to be the extremophiles. Earlier lecture, I mentioned these are the ones, North Pole, South Pole, everywhere in between. And I remember kind of reading where they think they may have isolated some of these archaea from a nuclear reactor, okay? Hot springs, bottom of the ocean and the sulfur springs, maybe a nuclear reactor. Having trouble finding that report. But these guys also are not parasites. They're not pathogens of plants. Why not? Because we're not extremophiles. These guys are going to be in the extreme environments. The majority of them are. You're not going to find any of us eukaryotes, any plants growing in places that are like that. Us eukaryote, plants, animals, fungi. First, there were the unicellular, the small, single-cell things. And then over time, millions, hundreds of millions of years, we began to develop where the single unicellular things started showing cooperativity. Remember the early sponges. Think back to your biodiversity. What was cooperativity started to become truly working together, started to get, almost what we would see as multicellular organisms. 6 kingdoms, okay, not phyla here, 6 kingdoms. Vary in shape, size, physiology, all across the board. We're not quite the extremophiles, but you can still find eukaryotes, North Pole, South Pole, most places in between. And then the current bane of our existence, the viruses. As we're going to see throughout this semester when we talk viruses, these guys are obligate parasites. They are non-cellular organisms. Okay, there's no plasma membrane, no organelles. We don't even think of them in the terms of prokaryote and eukaryote. They don't have a cell. They are obligate parasites because they have to be in us eukaryotes, in the prokaryotes. They have to infect a cell. They hijack the metabolism of their host cell, turn that host cell into a virus-making machine. One goes in, hundreds, if not thousands of viruses come out. Their genomes, DNA, RNA, single-stranded, double-stranded, linear, segmented, and everything in between.